Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Matthew B. Frieman. He's an associate professor at University of Maryland School of Medicine in the microbiology and immunology area. And he has his own research, which we'll get into. And he's also here to participate in the virus book that I'm putting together. So, Matt, thanks for coming.
Thanks for having me. Happy to be here.
Yeah. Well, if you would tell me about your research, what's that about first?
So my research is on coronaviruses. So we're taping this in the middle of a coronavirus pandemic. And it just so happens that I've been working on coronaviruses since around 2004, when I started my postdoc at the University of North Carolina in Chapel Hill. And so there I started on SARS-1, which we now call it SARS-1, or generally called SARS, working on pathogenesis and host factors and viral proteins that affected disease. And then I started at University of Maryland School of Medicine in Baltimore in 2009 when I started my own lab, again, still working on SARS. And then in 2012, we worked on MERS coronavirus, which emerged then. And then now merging that work into SARS coronavirus 2, where we're trying to study all aspects of therapeutic design, pathogenesis, how the virus works, causes disease, replicates, and really trying to understand a bit more about the virus than we already do.
Well, I guess to start off with, I have an interesting question. I keep hearing that, SARS-CoV-2 binds to this ACE2 receptor on a cell, and but when I look in the literature, I don't literally see a step-by-step, virus approaching the cell. And all the entry steps, every single one of them. Do you know if that's been elucidated, or are they just seeing, the last step, I guess, the ACE2, binding and entry? And is it, have they backed it up with electron, microscopy yet, or what's it look like?
Oh, yeah. So we have a lot of information about that now. There's really amazing cryo-EM and crystallography pictures of the spike protein on the surface of the virus binding to the ACE2 receptor on cells. There is a lot of understanding now about how the spike protein, which is the surface protein on the virus, how that gets cleaved by surface proteases, one called TMPRSS2 or TMPRSS2. There's also a protease called furin, which is important for cleavage of spike as well, and really how that entry mechanism works as the virus gets inside the cell. So we know a bit more about, every one of these coronaviruses. Every time there's a new one, and more people start working on it, we learn a little bit more about how this kind of all the little tricks that these viruses use to get into cells. So we certainly do know it binds ACE2, and that's important for its entry. And there's actually one of the important ways that we're developing therapeutics. A lot of people are doing this, where we're looking at drugs and other proteins that can block the entry mechanism of the virus into cells as a way to limit its spread inside animals and then eventually people.
If you would, just mechanically, literally, step by step, what happens when when SARS-CoV-2 approaches a cell? Does it have like a tail fiber that brushes against the ACE2 receptor, or like how does it work step by step? If you don't mind, if you know.
Sure. So it's not really a tail fiber per se, like phage has these fibers, or adenovirus has these fibers on the surface. So the coronaviruses have what we call spike proteins. They kind of look like lollipops on the surface of a tennis ball. And so these spike proteins have a long stalk as their kind of major arm. They're stuck in the membrane of the virion. And then the tip of this lollipop is called the receptor binding domain. And that's the region that binds to the receptor on cells. And one of the interesting things about SARS coronavirus 2, which SARS coronavirus 1 does not have, is it has something called a furin cleavage site, which is a protease that is important for a bunch of things inside of a cell, and it cleaves proteins in all aspects of kind of cell biology. But what we know is that the cleavage of this spike protein by furin, also by another protease that's called TMPRSS2, is really important. And so what seems to be happening is that the spike protein can bind to the ACE2 on the surface of cells that express it. So in your lungs, this is the ciliated epithelial cells in your lungs. These are the ones that they have these long hairs on the surface of the cells that they beat back and forth, and they mix their job is to mix around the mucus that your goblet cells, which are right next door, they make in lining your airways. So they keep your airway nice and wet so they don't stick together when you breathe. And only on the surface of the ciliated cells do you see this protein called ACE2, angiotensin converting enzyme 2. And so the virus spike protein with this lollipop on the end sticks to the ACE2 on the surface of cells. Right next to it is also on the membrane is also this TMPRSS2 protease. And so what we think goes on, and we're pretty sure is that there's actually two different ways the virus can get into a cell. So once the virus binds to ACE2, it somehow, a really unknown signal event happens, and you get internalization of the virus bound to ACE2, internalizing into a vesicle, so a little bleb into the cell, and that fuses to a endosome, which is this kind of entry vesicle that is inside of the cell. Inside the endosome are a bunch of other proteases, and those proteases can cleave the spike protein, which allows this really large conformational change to occur, and then that shoves the spike protein in this new fusion state into the membrane, and then you have kind of like two soap bubbles mixing together. They fuse their membranes together, and now that releases the genomic content, the genomic RNA of the virus into the cell. That's the kind of the standard dogmatic entry mechanism. The other entry way that seems to be also happening at the same time is that when you're at the surface, the virus coordinates with the ACE2 protein. There's also this TMPRSS2 next door, right near it on the membrane. If you get cleavage outside of the cell on the surface by the TMPRSS2 protein, you can actually get fusion right at the membrane. So instead of going through the endosome in that pathway, you get fusion right at the membrane, which allows for the genomic RNA to be released right at the plasma membrane fusion event rather than going through the endosome. So those kind of the two basic ways that the virus gets into the cell.
So does the virus first bind to ACE2 and how? And then once it does it, is there another part of the virus, the virion, close enough to TMPRSS2? I mean, I would guess it'd have to be within nanometers, tens of nanometers away, like another surface feature. Where do these surface features migrate? Like, between these two receptors or surface features, how do they feel, like physically, literally, how do you imagine that they interact?
Yeah, what we know is that if you have a cell that it does not express ACE2, but expresses TMPRSS2, you can't really get very good infection. So you need ACE2 as its main receptor. If you have a cell which expresses ACE2 and then also expresses a lot of TMPRSS2, you can drive the entry through this plasma membrane fusion event rather than going through the endosome. And so there are drugs like hydroxychloroquine, which has been in the news a lot at the beginning of this outbreak, which it inactivates the endosomal pathway by affecting the endosome, acidification of the endosome. And even in cells that express both TMPRSS2 and ACE2, you can actually still get infection because the virus it doesn't need the endosomal pathway. It can only use the membrane fusion pathway by TMPRSS2 cleavage. And so what happens on the surface of the cell is that we believe the ACE2 and the TMPRSS2 are quite close. And so as the virus spike protein kind of docks and binds to the ACE2 protein, the TMPRSS2 is right there, and it can cleave probably the same spike protein that is now docked to ACE2. It could be another spike that's there. There's not evidence yet, I think, that you have to have binding for the cleavage to happen, but it at least has to be really close, so close enough. And there's dozens of spikes on the surface of virion, and so you get this coordinated interaction where it's not really just one spike, but it's multiple spikes binding to multiple ACE2s and being cleaved at the same time by TMPRSS2. So all these things are kind of happening at the same space on the surface of a membrane.
If I'm just going to label like spike number 1 is the one that binds to the ACE2, and spike number 2 is the one that get caught up by TMPRSS2 nearby. Do you think that an interaction with spike 2 would then transfer, let's say, through the virion's capsid and affect spike 1, or do you think it literally has to affect the same spike protein in order to cleave it, or do you think that again there's this cross capsid action that could happen?
Yeah, so there's no capsid in this virus like it is in rhino or norovirus, where there's like a candy shell, this capsid on the outside. This is just it's a fluid enveloped virus, so it's a membrane that all these spike proteins are stuck in. There's a couple other proteins on the membrane also, E and M envelope and membrane protein, but it's there's not a capsid per se. So there doesn't seem to be a coordination between the spikes. They all just are kind of fluid and next to each other. And in their direct interactions with the membrane, there's clearly multiple ACE2s that are coordinating the same virion, and clearly multiple TMPRSS2s that are cleaving at the same time. So I think this is all kind of happening in more of a high affinity, but also high avidity. So you get multiple spikes binding at the same time that coordinate this interaction.
Okay. So you think it takes multiple spikes in order to have a successful entry? And, if you compare the endosomal version versus this, the outer membrane fusion version, which one appears to be less energetically, or easier to do, I guess, or more likely to happen?
So I think I would say there's almost definitely has to be multiple spikes that engage. There's going to be some on and off rate between the spike and the ACE2 interaction. And that off rate is high enough that, the virus could, fall off basically if there's not more than one ACE2 and more than one spike binding to each other. And also when you enter the cell, there's a lot of other cellular proteins on the cytoplasmic side that cause this curvature of the membrane. And you need this integration and signaling basically from the ACE2 to the inside of the cell that there's something there to pull and to cause this invagination. And so there's clearly going to be multiple ACE2s that are ongoing at the same time for these spike interactions that the cell knows there's something to pull on, if that makes any sense, and bring into the cell. Whether one is more important than the other, it's really not known. I will say that if you have a mouse in this case where this was all done for SARS-1, but if you have a mouse that doesn't express ACE2 at all, it's deleted for the ACE2 gene, you have no infection at all. But if you have a mouse that has the mouse ACE2 there, but you delete the TMPRSS2 protease, you still get infection and disease. So the ACE2 is clearly the major binding partner. There's also some carbohydrate linkages there that seem to be important, but that they kind of enhance the binding. But the TMPRSS2 clearly plays some role. And it's actually a really good therapeutic target now, where there are drugs called, there's a drug called Camostat and a drug called Nafamostat that target and inhibit the TMPRSS2 protease that are being used in vitro and in vivo in animals and clinical trials to see how well it could work against SARS-2 and COVID-19.
Of the various cell types we have in our body, from what I've heard, a lot of them express ACE2, but how many express TMPRSS2 and in a close enough range to the ACE2 receptor that they could be candidates for entry? Maybe it's better to target TMPRSS2. Maybe it's expressed less often on less cell types.
Yeah, absolutely. So there certainly is a lot of cells that express ACE2. This clearly is a respiratory virus, and all the coronaviruses are, I guess, other than feline coronavirus, which is a gut infection. And so there are, the ACE2, TMPRSS2 is pretty universally expressed in a lot of places. ACE2 is a little more restricted. So all of these things kind of go together in the way that they're expressed in the types of cells. There's been a lot of single cell sequencing going on where they're looking for cells that express one or the other, either TMPRSS2 or ACE2 or together. And they usually are generally expressed together in most places. The heart doesn't express TMPRSS2 very much. It expresses a lot of ACE2 because ACE2 is involved in blood pressure regulation. There are some cells that express more TMPRSS2, like kidney, that have less ACE2, but in the nasal epithelia, in the lung, in some parts of the gut, in the ileum and the colon, they express both ACE2 and TMPRSS2. And so, there are other potential sites of infection in these other organs and locations. It's not only these proteins that allow for infection, though. So there's other host factors that are still being mapped that seem to be important for entry of the virus and replication that can also play a role. And so that we don't really see a lot of infection of SARS-1 in the heart. It was never really something that was seen. For SARS-2 now, we certainly are seeing some more heart issues in patients than SARS-1. Now, that could be that the people that are being affected by the virus, there's a much larger population, a larger number. So you're seeing people with other comorbidities and other pre-existing conditions that allow this to happen more than others. But the other aspect of this is that the spike protein of SARS-2 binds to ACE2 at about 10-fold better than SARS-1 does, 10 to 100-fold, depending on the kind of assay. And so even if you have low-level ACE2 expression, you might be able to get the virus in to cells that SARS-1 couldn't infect because just the binding kinetics weren't very good, where now SARS-2 can infect well. So all of these things are still being figured out at a all this is done at the in vitro level in a petri dish. We're still trying to figure out what this means in vivo, both in humans and in animals, to figure out how this differential expression of the receptors and also how the different affinity of the spike with the receptors differs between the different coronaviruses and how this really affects disease.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
And you mentioned there could easily be circumstances where multiple virions are, in various stages of fusing to a cell. Do you think that all it takes is one virion to enter a cell and you got infection? Or do you think it may take coordination of multiple virions? And if so, what kind of disease state would you expect the cell to have versus one virion infecting it?
One virion can easily infect a cell and replicate. If you get multiple virions in a cell, you bump up the amount of genome that you start with, so you probably get more virus faster. But certainly one virus can go into a cell and cause disease, and replicate and make a million particles, quite easily. So I don't think there isn't a requirement for multiple virions to be in the same cell.
Do you think it can happen, or is it, what if it's like sperm and egg model? As soon as one virion enters, the membrane changes in such a way that no more virions are allowed in. Has that been looked at, or is that not really important?
Yeah, that's been looked at. I mean, that isn't an issue. There are certain other viruses. I think pox viruses do that, where they turn on some kind of anti-pox effect, where one virus goes in and then it doesn't let the other ones go in by affecting the receptors on the surface. That doesn't seem to be true for coronas, as far as I know. One of the interesting things that coronaviruses do, though, is that when they do bind to ACE2, they internalize the receptor with them to go into the endosome. So if you have a lot of virus on the surface and you're getting multiple virions or a lot of virions in the cell, you can actually reduce the amount of ACE2 that's on your surface to basically reduce the function of ACE2 in the lungs in particular. And ACE2 is involved in repair of lung tissue. So one of the ideas is that in cells where there's a lot of virus and potentially a lot of spike that is being secreted, you can actually get a bigger reduction in ACE2 on the surface of cells. And maybe that is a part of the effect of how the virus causes so much disease. And so all of that was been predicted and demonstrated for SARS-1, that at some level that's important for SARS-2 as well. But clearly the virus is causing damage to the cells it infects and then altering the immune response around it to the neighboring cells. And so that's all part of this pathology that we're trying to understand both in animals and in humans.
So what do you think the cell is sensing? Why is it allowing the virions to enter? Do you think there's any discernment as to, okay, this is, just let it in because it hit this receptor regardless? Or again, is there discernment on any level?
Yeah, no difference that we know of, but that where ACE2, can't really decide what it binds to, and then it can't really decide how to transit that signal to the cell for internalization. That's certainly not known. The cell has a whole host of other ways of protecting itself from viruses that are kicked on once it recognizes that a virus comes in, but it doesn't seem to be at the level of entry. It seems to be at a level post that, after there's all of these innate immune factors that sense a virus is there or not there and try to fight it.
Okay. How long does it take? I don't know if it's even observable, but how long does it take for this, once the first moment of binding occurs to an ACE2 receptor or TMPRSS2, how long does this whole process take until the virus is inside of a cell?
Once it gets inside, it's a matter of minutes from basically binding to where the genome is in the cytoplasm, 15, 20 minutes maximum, probably faster than that. That's pretty much as fast as we can see it. But I would bet on a population-wide, it's, there's definitely faster cells that bring in the virus and then allow it to get into the cytoplasm.
And then how long do you think the delay is to, replication in the first lytic event? Once the cell is infected, you're saying it takes minutes. And then how long till the, a lytic event occurs in that cell? And do you think there's any coordination, any latency where enough cells have to be infected and then maybe there's a cell-to-cell communication that coordinates a lytic event? Or do they seem to just happen one after another? If cell A is infected at this moment, 4 to 6 minutes later or an hour later, it lyses, and cell B happened a minute later, so probably a minute later it'll lyse. Or is there any, any coordination? And is the period between the lytic event prescribed? Do you know anything about it?
So there's certainly some viruses that have latency. That's the wording you're using, where virus can basically go silent for a while and then it can be reactivated. So there's no latency in coronaviruses. There's no active lysis either, where there's not a factor in the cell, in the virus, that causes the cell to burst like a a lytic event like phage or some other coronaviruses do, or other types of viruses. Basically, the virus goes into the cell and it starts making more copies of itself. It doesn't want to kill the cell super fast because it wants to crank out as much as it can of making copies and making new virions. So, let's call it, let's say 10 minutes for the virus from it binding to ACE2 to getting it out of an endosome. It takes about an hour or so before you can start detecting viral RNA. It probably happens faster than that, but we can't see the first few rounds of that occurring. And then you have translation of the mRNAs that are made. Coronaviruses make these things called subgenomic RNAs, which are kind of cool ways of viruses to make new RNAs in the coronavirus family. They have this interesting way of kind of hopping the genome to add the very 5' end of the genome onto every single one of the RNAs that is there. It kind of acts as a promoter for translation. And then once translation happens, you start making your spike and your envelope and your M protein, which get trafficked through the ER and Golgi. They grab the genome, which has been, it's being made in these things called double membrane vesicles, which are these literally two membranes around in a vesicle that the virus induces by some of its replicase proteins to form in a cell. The new data actually shows that there's RNA inside these vesicles. No one really saw this before. It was always predicted, but no one really knew if it was true. So the virus kind of hides out its replication machinery inside these vesicles, and then it has a pore on the membrane of the DMV that allows the RNA out. And once it comes out into the cytoplasm, it gets immediately wrapped up by the nucleocapsid protein, kind of look like a histone or chromatin factor that you think of in DNA in the nucleus. And then the newly forming virions, as the structural proteins are leaving the ER and the Golgi, they coordinate with the N protein, the nucleocapsid wrapped around the genome, to internalize this new virion into essentially a vesicle that's going to leave the cell. And you get multiple virions fused into this vesicle, so it kind of looks like a bag of marbles. And then that fuses to the membrane and then releases the virions to the outside of the cell. And so the virus doesn't want the cell to die. It wants to be able to make as much of these virions as possible while overtaking all the machinery in the cell. So eventually you do get death, but it doesn't want it to happen super fast. It wants to be able to crank out as much virus as possible.
So the cell will start releasing viruses in bundles, but it won't necessarily die or kill itself from apoptosis for a while.
Right. So that's kind of the same way that the virus, the kind of soap bubble imagery, that you have the same soap bubble from the inside of the cell now fusing to the membrane, and that fusion event now kind of spills out the virions outside of the cell. And so now they're available outside of the cell, and if you're in the lung, you're in this kind of mucus layer, and then you get infection of the neighboring cells that are now, hopefully, at least for the virus side, receptive to being infection because they have ACE2 and all the other factors you need.
So what factors would then cause a cell to kill itself, to undergo apoptosis, or to be attacked by our immune system?
Yeah, this is the cool part about viruses, right? You can call them nature's cell biologists. People call them nature's bioterrorists. There are all different ways that viruses of all the families have to manipulate the cell to allow for them to do their job from the virus standpoint, which is to basically make more copies of itself. And so this is the stuff that we work on in lab, where all of us, basically working on these viruses from all kinds of families, try to figure out how it is that the virus goes and replicates in the cell, because the cell has all this other machinery that tries to shut the virus down at the same time that the virus is trying to replicate itself. And so it's this kind of back and forth. There's Harmeet Malik and calls this a viral arms race. And so there's this constant evolution, over long periods of time, of the virus making proteins that can basically break the signaling events that happen in a cell that the cell tries to fight back against the virus with. And so one of the major proteins in cells that viruses that cells have is called interferon. It is at the bottom of this pathway that is turned on when the cell recognizes that there's a virus in the cell. And so one of the major ways the virus knows that the cell knows there's a virus there is by detecting its RNA. So the cell knows what its RNA is supposed to look like. And if it detects other RNA that's there that doesn't look like it's an RNA because it has different beginning or an end, or it has different proteins on it, it can turn on the signaling event that turns on interferon. And then the cool thing about the interferon part is that it then gets secreted from cells. it can bind back on the same cell or on neighboring cells. And it's this alarm signal that tells everybody in the neighborhood, hey, there's a virus here. And so even in the uninfected cells, when interferon binds the interferon receptor on the surface of cells, and then it starts a huge cascade of signaling events inside the cell that allows for the cell to turn on these things called interferon stimulated genes, or ISGs. And these are hundreds of proteins that now basically prepare the cell for a new invading virus. And so that happens in the cells that are already making the virus, or the virus is infecting. And SARS and other viruses, but in particularly, SARS and the coronas, have proteins that block these pathways that the cell is trying to turn on to tell everybody that there's a virus there. So it blocks the interferon signaling pathway. It blocks the pathway that chews up the viral RNA. It blocks a bunch of kinases and nuclear import factors that would be normally turned on for the cell to recognize there's a virus there and to turn on all these interferon stimulated genes. And so there's this kind of back and forth that goes on all the time when viruses infect cells of what things the cell is responding to, what the virus is making, and basically how the virus can hold out long enough to make more copies of itself. And then the cell trying to hold out long enough before it dies or it gives a death signal to itself to try to kill the virus. And at some point, the cell kind of, gives up the ghost and says, okay, I'm going to turn on apoptosis and kill myself for the sake of the organism, if it's in an animal. And so you have this kind of back and forth tug between the virus and the host about all these different signaling events and factors that go on all the time.
So it seems like there's some deliberate signaling from the viruses inside of an infected cell. And then there's also some counter signaling by the cell itself, the cell itself saying, help, I'm infected. And then the virus is putting out material that will, niche construct other cells to prepare them or regulate them in such a way that they're ready for infection.
So it's not, I don't think there's ever, for the coronas, there doesn't seem to be a protein that's secreted by the cells from the virus to then kind of sensitize other cells to infection. But there certainly is protein that's secreted by cells to signal other cells that to kind of as a danger signal to make sure that they are prepared for kind of the coming onslaught of the virus that's gonna, in their vicinity.
Oh, okay. I was going to ask you, how are these instructions packaged? Is it extracellular vesicles that the cell gives off? Is that how the cell does its signaling? And if the virus is doing signaling, what does it look like? What comes out of the cell that's different?
From the cell side, the virus secretes interferon, which is a protein. So it gets made, and then just like a normal secreted protein gets the ER and Golgi. Really, at the same time, the virus is trying to make itself as well, and then it gets secreted through just a normal secretion vesicle that leaves the cell and goes and floats around outside of it to the neighboring cells. And so you have those. You have also things called exosomes, which are little vesicles that fuse and that are released from the membrane of the cell. And there's been shown in especially in the placenta and other cells where you get secretion of these exosomes. So it really like kind of a secreted vesicle that has which been shown in there to be a protein, but also microRNAs. And those can then fuse to neighboring cells and then bring those microRNAs from one cell to the other. And when those microRNAs get into cells, then they do what microRNAs do, which is then bind to RNA and then turn on or turn off genes in the neighboring cells. So they kind of act like these extra signaling molecules that are alerting, can affect the other cell's ability to respond, usually in an antiviral way. So it turns on proteins that are important for protection of virus. So kind of like interferon does. For the coronaviruses, there doesn't seem to be, at least that's been shown yet, any kind of definitive signaling or protein or RNA that's secreted from the infected cell to the neighboring cells that allows for alterations of the neighboring cells, other than really trying to limit the amount of interferon that's being produced from the infected cell.
But when these substances are released, they're packaged in exosomes or they're packaged in some sort of extracellular vesicle, right?
Right, exactly. Yeah, for the exosome side. Yeah. The protein side, they're just released from the cell like a normal secreted protein would be.
Oh, so the cell can release just naked proteins that are
absolutely.
going out
Yeah.
into the interstitium.
Yep, exactly.
And they have enough survivability that they can enter into other cells still? Or their purpose is not to enter into other cells, but then they're picked up by outside factors, the immune system, et cetera.
So for interferon, so then there are interferon receptors that are on the surface of other cells. And so just kind of like ACE2 is binding to virus, you have all of these other the cells are covered in proteins that are sensing the external environment to get external signaling or stimuli, whether it's growth factors, whether it's interferon and immune signaling proteins, all kinds of things. And so the interferon will find its cognate receptor, so this interferon receptor on the neighboring cells. And then once that binding happens, then you have a signaling event that goes inside of the cell to turn on all of these interferon stimulated genes in the neighboring cells. So there's a huge kinase pathway that gets turned on when interferon or different types of interferon are bind to different interferon receptors on the neighboring cells.
Okay. Why do you think that, for instance, SARS-CoV-2 has such a long period of time between initial exposure and, pathogenicity? Seems like, several days, it could be longer, maybe up to 2 weeks. Why this latency period versus, a few hours?
So you have a lot of things going on at the same time. So SARS infects a cell. One, it can start replicating. You have this kind of arms race going back and forth. So the cell trying to diminish the amount of replication versus the virus trying to replicate as much as it can. So you have that kind of level of back and forth going on at the very early stages of infection. The virus is able to replicate without really being detected. So it expresses all of these interferon antagonists and other host factor antagonists that kind of manipulate the cells it infects to allow for it to hide out. So you don't feel sick early on, potentially, because the virus is able to manipulate your immune response, which is the thing that makes you feel sick. And so if it diminishes this early response, the virus can essentially hide out for a while, replicating without being noticed and without your body, your lungs knowing the virus is there. And then once you get this overwhelming response, because now all of a sudden the virus, the host, your lungs and your immune response realizes that, oh no, there's a ton of virus here. What's going on? And so it makes this huge immune response back at the virus. And that actually causes more disease than you would normally see if it was a kind of a balanced response. So we call it a dysregulated immune response when the host recognizes that there's all this virus there and it didn't know about it. And so those early stages can take a while for the virus to go and to take off. So you have kind of both ways of this. You have the virus kind of replicating slow if there's all these host factors that are slowing it down, and you have it hiding out for a while before you get this big damage response that occurs in the lungs after there's virus there that's replicating and kind of going crazy.
So what other breakthroughs of understanding of SARS-CoV-2 do you feel like either you're close on or you're seeing other scientists are investigating? Is it figuring out exactly all the possible sites that could confound the spike protein, or what are like the really heavy areas of research right now?
So I have to say that the SARS-2 research is going at a remarkable pace. And so I would say every day there's new insights that are coming. From the therapeutics standpoint, one of the kind of interesting things is that there's been a huge amount of mapping of antibodies that bind to the spike protein. And so there's a lot there. We know the crystal structure of the spike protein. There's been a lot of crystal structures of antibodies bound to spike. And we know different types of different sets of antibodies, different individual monoclonal antibodies that recognize different aspects of the spike protein to block infection. So in that sense, from the kind of antibody side, it's going to be interesting to start combining these antibodies together to kind of get dual effects and dual ways of blocking the virus. So the virus can't just evolve away from one; it needs different aspects of its response to come back from. I think the kind of remarkable thing to me, just kind of broadly, is that with the number of people that are affected by this virus, we're seeing really a huge variation in response of disease. I find that to just be amazing. It's all the things that we talk about, that when a naive population is infected by a virus, you see this huge variation in disease. And just to see it play out, I find to be absolutely remarkable. Everything from just losing your, either not having any symptoms at all, so completely asymptomatic, to people losing their sense of smell and taste because you get infection of the epithelia in the nasal cavity in your nose. And then what we think is going on is you get death of the cells around the neurons. The neurons themselves aren't dying, but it's the cells that wrap around the neurons seem to be dying from the infection. And so that eliminates the signaling capability. So eventually the smell and taste comes back, but it takes a while for these cells to regenerate. So you have that at the kind of really kind of easy to get over phase, all the way through people having super severe, it's being obviously a lethal infection, but really widespread organ damage in across people. I find that to be really kind of remarkable on the kind of the pathogenesis side of this, that this virus, which is really quite similar to a seasonal virus that gives you a cold, all of a sudden, is able to cause this wide spectrum of disease. I find that to be really amazing. And it's really one of the things we're trying to figure out in the lab is how do these other comorbidities that are involved with, that all of us have internally, just immune response differences or physiological differences, how do those comorbidities affect the disease progression? And can we understand that from in an animal, and how does that reflect what we see in humans? So one of the things we're doing in my lab is, and we did this for MERS as well, is we're looking at diabetes and obesity and other comorbidity factors in humans, or in this case, mice, that can exacerbate disease, and to see what it is about being diabetic, or what it is about being obese, or what it is about it being immunosuppressed that alters the trajectory of this disease. And so that, from the animal side, that's really one of the major focal points of the lab now. And the other thing is, there's been a huge amount of therapeutics being developed, both things that are being repurposed from FDA-approved drugs that are on the shelf, as well as new drugs being developed. And we're doing a lot of repurposed drug screening now and testing in animals with a bunch of collaborators, as well as trying to develop new drugs in the lab that target host factors that are involved in multiple viruses for replication. And so it really, all of it is quite phenomenal to see all happening at the same time. And the goal from all of this, one, is to develop therapeutics for SARS-2, but two is to develop therapeutics for a wide variety of viruses that are broadly applicable and broadly antiviral, so that the next virus that comes out as an epidemic, things that we don't even know exist yet, we have therapeutics on the shelf for them. And I think we're limited in the amount of therapeutics we have, and for these emerging viruses, and we'd have to be better prepared next time.
What do you think happens or has been happening to, SARS-CoV-2? I mean, if I label the first person that got it originally as number 1, and they gave it to number 2, and they gave it to number 3, how many passaging events do you think guests on average, the average person will experience at this point? Like, if I get it tomorrow from someone, will I be number 13 or 50 or 80? Like, what's your guess in the passaging chain? And how do you think the virus would have changed by now, or has changed by now? Or if not, by passaging number 11 or 500, how do you think it'll change?
Yeah, so that's a great question. I think if you got it now, it really kind of depends on where you are. If we're in the US, we're probably, I don't know how many people it's gone through before it's got to you. It's an interesting question. Probably, I couldn't know. I don't know if it'd be 50, it'd be 500. I don't know where we would be yet. But we certainly have a lot of data on the sequence of the virus and how it has changed since it was initially identified at the end of 2019 in those first people in the fish market in Wuhan. And we know a couple things so far. One is that there's a mutation that's in the spike protein, which seems to be important for transmission. And it has this one amino acid change has allowed the virus to transmit better between people. So that's certainly there. The really dramatic thing for me is that from the very beginning, and it kind of says something interesting, I think, about where the virus, the timing of where we're seeing the virus, but is that from the very beginning, this virus was replicating very well in humans. So right inside, if you go all the way back to the December 30th, where we first identified or first heard about there being this fish market in Wuhan that was had these cases of pneumonia. If you look at the virus that was in those people that were infected early on, it really is not that different than what we're seeing now. So the virus hasn't had to mutate a huge amount for it to replicate and spread between people. And so what I think that means is that, and we've been saying this early on, and I think it's still now true, is that the virus had clearly been replicating for a while in people before we knew it. It doesn't mean it's replicating for years, but it clearly was replicating for weeks to months, where it jumped from animals to people, and then it went around at least a small population, at least from person to person, before we really knew it was there. Because it replicates, it didn't have to adapt from animals to people, at least, or from person to person from this early stages to spread really well. And what we know from SARS-1 and from MERS is that there has to be these evolving events that occur from animals to people for these viruses to replicate and be able to transmit very well from person to person. So there are multiple events that have to happen in the genome of the virus, especially in the spike protein and the way it engages ACE2, for it to go from animal replication to human replication. And so it's kind of a slow burn from animals to people, going back and forth as it mutates a bit before it really can take off in the human population. Even once it goes from person to person early on, it doesn't replicate that well, at least in SARS-1 and for MERS, and before it can transmit really well and replicate really, very well in humans. But this virus, at least the first steps that we knew about from these patients in Wuhan, it's replicated quite well. And so it clearly had been in humans for a bit before we knew about it from the beginning. Since then, there really hasn't been a huge amount of mutations. I mean, there are mutations that occur. You can find them by sequence. You can identify these small variants and look at them on phylogenetic trees to differentiate them. But there isn't at least a pathological difference that the viruses early on caused one type of disease and now they cause something different. There doesn't seem to be that trajectory yet. We haven't really achieved that, even with the number of people that this virus has gone through.
So it seems like viruses a lot of them seem to get out there, maybe have an outbreak, and then kind of fade away. Why do you think that is? Do you think that a lot of viruses, I guess, they become more commensal with their hosts, or why do you think that is? And viruses we haven't really been able to fix.
No, I think every virus has its own way, right? I mean, I tell all the grad students that that in the lab, when they're looking for projects and when they're worrying about a career, I tell them that everybody finds their way through this, and everybody will be okay. I think viruses have the same kind of thing for these emerging viruses, at least. Some of them come in and they burn bright, they replicate incredibly well, they cause a super amount of disease in people, and then they go away. And so, I mean, a perfect example is SARS-1. So SARS-1 emerged in late 2002 and 2003, and then it went away in 9 months. And one of the really dramatic differences in the disease between SARS-1 and SARS-2, even though they're really very similar viruses, at least genetically, is that almost everyone with SARS-1 that got infected had symptoms within 48 hours. And so it came into your lungs, you got really sick, you had symptoms, regular cough and cold symptoms, and then you were identified as being positive, as having symptoms, and you either got quarantined at home or you got sick enough that you went to a hospital and you got quarantined there. With SARS-2, its trajectory is quite different. We know now that there is, 5 to 10 days after you're exposed before you have symptoms, if you're going to have symptoms. And during that period, probably around day 5 to 10 in that range, you can be shedding virus, but you don't know it. And that's the reason that this virus took off and is spreading around so easily, and why everyone's supposed to wear masks when they go outside and around people, is because you can be infected and be shedding virus, but not know you're sick because your body hasn't presenting with any symptoms yet. And so just those two viruses, they're in the same family, they're very similar. This dramatic difference in the clinical presentation early on that differentiates them. And so SARS-1 didn't go away because we had some magical drug or antibody or vaccine for it in those 9 months. It went away because of really good public health measures that everybody who was infected had symptoms, and then you shut down the connection and the transmission events because those people were quarantined. For SARS-2, the shutdowns, at least in the US, the goal was to do that, where you eliminate this transmission. And then all the shutdowns and the quarantines measures were basically reversed pretty quickly, and there was still a lot of virus out there. And so from the virus standpoint is to find people to infect and replicate and spread. And so viruses do what viruses do, and that's what it did.
Well, I mean, you have the case of Sweden, and then in Europe, various places shut down, and in some there may be somewhat of a resurgence, and some not. I don't know. It seems like it's not just the one issue, but there's more to it. If you look also, let's say, at Ebola too, it seems to have peaked and then gone away. And I don't know, in Africa, I don't know if there was definitely strict conditions met to stop its spread. So do you think there's any other method of action besides just human containment that slows down the viruses or changes their nature?
Oh, sure. I mean, Ebola is a very different virus. Ebola has to be spread by bloodborne transmission. So the way of transmitting Ebola is much different than you're transmitting a respiratory virus. Every virus has its own way of spreading and its own issues of the ways that it's causing disease. And I think that, for Ebola, you had we know that it's bloodborne. We know that if you limit contact with contaminated blood, you can eliminate infection. We know that during Ebola, there was burial practices where they bathed the body as a kind of religious ritual, and that leads to transmission. So once that was blocked, you eliminate those avenues of transmission. All of those issues are different between a bloodborne virus, a mosquito-borne virus, respiratory virus. So there's different reasons why the viruses go away for all these conditions. I think what we're seeing in Europe now is that they're actually, all of these, there's a huge number of surge of cases all over Europe right now because the transmission event, they weren't totally blocked. And while they released quarantine and they tried to have all these measures of protection, it wasn't really able to eliminate the virus completely. And so when people stop wearing masks, they stop social distancing, you can now transmit respiratory viruses quite easily, like we do in the winter when we get a cold. And so we're seeing all this transmission all over Europe now. And they're now starting to have lockdowns in France and England and Italy and all of the places where they tried to basically reduce these measures in the last couple of months. And in the US, we're a perfect example where there's been most of the country now has some level of release of quarantine measures and distancing. There's mask mandates, either allowing masks to be worn and encouraging it, or encouraging people not to wear masks depending on what state you're in. I think what we're going to see is in these states in the United States where there is hotspots now, we're going to see that there is going to be a huge increase in these hotspots over the winter where people are mixing together and not distancing and not wearing masks and not doing kind of the good public health measures that are being encouraged. So, I mean, in the context of different virus families, every one of these has its own way of spreading, and you have to eliminate those transmission dynamics to slow them down. And we certainly haven't been very good in the United States about doing this.
Well, very good, Matt. What's the best way to find out more about your research? Where can people go?
Sure. You can Google me. You can go to the University of Maryland website and search for Matt Frieman. We have a little page there that kind of says what we do. And I'm also on Twitter. You can search Matt Frieman there. So I'm around a couple of places. And if you need to find me, I'm here for answering questions.
Excellent. Well, Matt, thank you for coming on the podcast. I appreciate it.
Sure. Thank you for having me.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.